Hi, hope you are staying safe at home in this critical time. Today I'm going to talk about a convenient strategy which can be used to find a drug for COVID-19. Here I start with the first slide, which is titled to COVID-19 pandemic. The most important things is to explain which method we should consider. First, let me explain you something about COVID-19 disease. It is caused by severe acute respiratory syndrome coronavirus. The outbreak was identified in Wuhan, China in December 2019, and common symptoms include fever, cough, fatigue, shortness of breath, and loss of smell. So far, 3.52 million people have been confirmed to be affected by COVID-19. 1.13 million people recovered and 248,000 were died. As we can see on the screen, fever is the most common symptoms and lungs are the most affected organ. By COVID-19, this is because the host cell, alveolar cell, are mostly located in lungs. The virus uses a special surface to connect and enter inside host cell. Currently, there is no any drug and treatment option, especially for COVID-19 disease. But to overcome the symptoms, some traditional medication like ibuprofen can be used. WHO, which is stand for World Health Organization, and NIH, which is stand for U.S. National Institute of Health, they don't disagree to use of anti-inflammatory drug like ibuprofen or aspirin. Uh, Here I'm going to show an overview of stages since a drug can be found until it's available in market. For the first stage, which I will discuss more, is discovery and development. The second stage is preclinical research, which candidate drugs should be used for in vitro and in vivo tests to find safety and efficacy of the drug, some other property like absorption, toxicity, bioavailability, and so on. At the third stage, researchers move to clinical centers and test the drugs on volunteer stage. On volunteer. In stage 4, FDA must approve the drug and let the company to produce. FDA means Food and Drug Administration of United States, and finally the products are required to be monitored. The stage of drug discovery is working like cycle. After collection of the drug, primary and secondary assay, we can use direct and indirect design. These two methods are based on computer modeling, and I will explain more in the next slide. After we find candidate drug computationally, we can synthesize and apply for primary and secondary assay again for further preclinical research. Drug design can be either indirect, which called ligand-based, or direct, called structure-based. In ligand-based, the receptor, like protein, DNA, is unknown. We don't have any information about the structures and crystal structures. For example, QSAR, which use a set number of compounds, and IC50 to find a relationship between a structure of drugs and biological activity, their biological activity. In structure-based methods, we have receptor, we have information about the receptors, and we try to find the best drug which can be interact with active site. And here we can see the protein structure of COVID-19. The main protease, the crystal structure was reported in Nature paper with collaborating between Shanghai Technology University, Cancer Center Cleveland, USA, and University of Queensland, Australia. So here we have the receptor, we have information about it. As I said, we can follow a structure-based drug design method. My suggestion to start this method, as we don't have any initial drug structure, we can use compound library, and since there is a similarity between COVID-19 and SARS, we can start with corresponding drug which is used for SARS. And next we can apply for molecular docking. Molecular docking is on computer modeling and show which compound can fit better in active site. Protein have active site, and these active sites are responsible for biological activity of proteins. And next slide. Moreover, we can run molecular dynamic simulation (MD). MD simulation gives more information like interactions, energies, stability, while the protein and drugs together in complex form are in solvent. Also, we can set them in a different temperature and pressure. All these movements of atoms that you are watching on animation form is following the first equation of Newton's laws of motion, and it gives more natural system. Okay, so far I explained the strategy of a structure-based drug design method to find lead structure, to find the lead drug for COVID-19. Here we move to clinical research because computational work is not enough, even though computational work is time saving and chemical saving, but it might be different in real condition. Preclinical research can be divided in two methods: in vitro and in vivo. In vitro is experimental model of drug within the glass, and the drug has not a rule in fasting diet, body temperature, and so on. It's cheaper, less precise, and quick result. In contrast, in vitro means experimental models of the drug within living cell. The drug has a rule in fasting diet, body temperature, and so on. It's more expensive, more precise, and time consuming. Uh, at the end of my presentation, I'm going to close my speech with this example. There is no any fixed strategy in research, and we should face many failure until to reach success. This is also same in our life. Hope you are staying safe at home in this critical time of COVID-19 pandemic. Thank you so much.